Nature is such a great thing that there is much variation in all respects, such as their structures, functions, and modes of reproduction in different organisms. I myself, Dr. Mangal Patwardhan, head Department of Botany, Gokte Joglekar College, Ratnagiri, is going to discuss with you one of the important feature of nature, and that is sex determination. This is part 1 of my video. Actually, every living entity aims to reproduce by various methods such as vegetative, asexual, and sexual mode of reproduction. In primitive organisms, we can get the vegetative mode of reproduction as well as asexual mode of reproduction. But the ability to reproduce sexually is said to be the evolved character. Uni and multicellular organisms reproduce sexually, and generally male and female sex organs develop in the animals as well as plants. In animals, we can get various sex organs, and in plants, flower is the sexual reproductive part of the plant where we can get different sex organs. Male sex organ is androecium and female sex organ is the gynoecium. So androecium consists of different anthers or stamens, and in the anther lobe we are getting the development of pollen grains, while in gynoecium there are ovary, style, and stigma. These are the three parts where the stigma is said to be the receptive organ or the landing phase for the pollen grain, and through style the pollen tube comes down to fuse with the ovule where egg cell is situated, and hence there will be the fertilization process affected, and thereafter there will be the development of the embryo and the seed, and seed will be useful for the further propagation of the plant. There are various types of organisms where the organism may be unisexual or dioecious, where there are two separate houses or bodies developed for two different types of sex organs, male and female. There are certain organisms which are bisexual or said to be the monoecious, where male and female sex organs are produced on one and the same organism, while there are certain organisms which are said to be hermaphrodite, where production of both eggs and sperms by the same organism is there. This is the case with lower animals. In dioecious organisms, the two sexes may be distinguished only on the basis of primary characters or secondary characters. The mechanism of sex determination. The mechanism of sex determination may be genotypic or environmental. Genotypic is abbreviated as GSD. It is nothing but the genetic or chromosomal type of sex determination, while environmental sex determination is abbreviated as ESD. Today we are going to study the chromosomal sex determination. For this purpose, first we will consider the types of chromosomes. There are two types of chromosomes in the living organisms. First one is the autosomes, and for autosomes, A, capital letter A is taken. A is a haploid set of chromosomes, while there are sex chromosomes, and the sex chromosomes are responsible for determination of sex. Autosomes are responsible for fixing the vegetative characters of the organism. In the chromosomal mode of sex determination, that is, based on the type of gametes produced, there are various modes of sex determination. First mode is the heterogametic males. Second one, that is the heterogametic females. And the third one is the haplodiploidy. In heterogametic males, the patterns XX, XY and XX, X0 are present, while in heterogametic female there are ZW and ZZ mode, and ZW Z0 mode is present. Now we will discuss the details of the heterogametic males. In this case, the male produces two types of gametes and female is going to produce only one type of gametes. So now first we will see the type 1 of heterogametic males, that is XXXY mechanism of sex determination. This type is commonly seen in mammals, in insects like Drosophila, and the plants like Spirocarpus, which is a bryophyte, and Melandrium. Here XX female is going to produce egg and XY are males going to produce sperms. In these organisms, the female, which is a homogametic in sex, produce only one type of gamete and has the XX genome configuration along with the autosomes A. So in that case, total configuration will be AAXX, and the male being heterogametic produce two types of gametes, one containing X chromosome in addition to the autosomes and the other containing Y chromosome in addition to the autosomes. The male has the XY genome configuration along with the autosomes, that is AAXY in diploid cells. In Melandrium and in mammals, the presence of Y chromosome is required for the development of a male sex phenotype, but in Drosophila, the Y chromosome plays no significant role in sex determination. In mammals, the X chromosome present in any number in absence of Y chromosome gives rise to the female sex phenotype. The presence of a single Y chromosome, even in XXXY, gives rise to the male sex phenotype. The Y chromosome includes the development of undifferentiated gonadal medulla to develop into testes while the XX chromosome complement, or rather in absence of Y chromosome, includes the differential gonadal cortex of ovatestes to develop ovaries. The XRY gene present in sex determining region in the region 1A of short arm of Y chromosome in human being triggers the developmental undifferentiated gonadal tissue of the embryo to form testes. The product of SRY genes called as test determining factor, that is TDF, is also present in adult male testes. It exhibits a very dominating effect on the development of the sex phenotype even in presence of 3 or more X chromosomes. Therefore, in parents, we can get 44 plus XY complement in males, and in females 44 plus XX. It produces the sperms, and when we develop the checkerboard, we can observe males having 22 plus X and 22 plus Y, two types of gametes, and in females 22 plus X, monotypic gamete. The result will be 44 plus XX and 44 plus XY. Thus, the 50% males and 50% females occur as a result of XXXY mechanism of sex determination. Now we'll consider the second mechanism in heterogametic males, that is XXX0 mechanism. The pattern of sex determination is commonly observed in grasshoppers and bugs. In this pattern, the female possesses XX genotype and produces identical gametes. The male carries only one X chromosome and no Y chromosome, or no other chromosome is present. In plants like Dioscorea and Vallisneria spiralis, we can observe the same type of mechanism. The male carries only one chromosome. As a result, half of the gametes are with X chromosome and the other half without X chromosome. In this case, if we observe the parents, in male parent, the composition is AAXO, while in females, AAXX. If we develop the checkerboard, two types of sperms, A plus X and A plus 0, that is without second sex chromosome, males produce two types of gametes, and females there are homotypic gamete, that is A plus X. As a result of fertilization, an egg AX gets fertilized by a sperm carrying AX chromosome will result in female, while fertilization by a sperm with A0 chromosomes will result in males. Thus, here also we can get 50% males and 50% females produced naturally. Then second type is heterogametic females. Among the heterogametic females, two patterns of sex determination are known. First one is the ZWZZ mechanism. The two sexes differ in chromosome morphology, and ZOZZ, where two sexes differ in chromosome number. In these organisms, male is homogametic sex. In order to avoid confusion, the sex chromosomes are named as Z and W instead of X and Y. So now first we'll discuss type 1, that is ZWZZ mechanism. This system is common in birds, reptiles, fishes and insects. The diploid female carries one Z chromosome and one W chromosome in addition to the two sets of autosomes. So the composition of diploid female will be 2A plus ZW and males 2A plus ZZ. Thus, 50% of the gametes produced by females carry Z chromosome in addition to a set of autosome, and the remaining 50% gametes contain W chromosome in addition to the set of autosomes. So as a result, we will get 50% males and 50% females produced naturally. Z, Z, Z0 mechanism of sex determination is the second type in heterogametic females. Butterflies and certain moths exhibit ZZZO mechanism of sex determination. In plants, we can observe it in Fragaris. The genotype of diploid female carries a single Z chromosome and two sets of autosomes. The genome composition leads to the production of two types of female gametes and one type having a Z chromosome in addition to a set of autosomes, and the other carries only a set of autosomes. The male is homogametic and produces gametes with a set of autosomes and a Z chromosome. So parents are of two types, AAZO female and AAZZ male. Again, in the checkerboard we can observe 50% males and 50% female individuals are produced as a result naturally. Now we'll discuss about the third type, that is haplodiploidy and sex determination, which is present in the Hymenoptera. Especially we can observe the honeybees are showing this type of mechanism. In the order Hymenoptera honeybees, the male, that is drones, develop from unfertilized egg and have a haploid chromosome number, that is 16. While in females, queens and workers, they arise from the fertilized eggs and hence they are diploid and showing 32 chromosomes. Egg haploid plus sperm haploid develop a diploid status present in females, and females may be either a queen or workers. Second type, eggs haploid, they will not fertilize. Then the sperms, they will not fertilize and they produce haploid drones. It has been shown by Whiting that heterozygous, homozygous and hemizygous status of certain chromosomes control the sex determination and not the haplodiploidy. Female determination depends upon heterozygosity for a part of a chromosome. Hemizygous individuals cannot be heterozygous and therefore males. That homozygous diploid males can be obtained. Thus, in this part 1 of my video, we have discussed heterogametic males, heterogametic females, and haplodiploidy type of sex determination. Thank you.